Okay, all right. So ayan, hello dears. Okay, all right. So again, welcome to the continuation of our pre-recorded lecture on concentration techniques for stool specimens. Okay, all right. And from the previous lecture, we've started to discuss the first type, which are your flotation methods. Starting with the first technique, which is your brine flotation method. And for this video, we'll start now with the second technique, which is your zinc sulfate flotation method. All right, so this method was originally developed by Faust in 1938 for the recovery of both helminth eggs and larvae and even protozoan cysts. Okay, the main reagent is again 33% zinc sulfate solution, which is the most widely used reagent for flotation methods. And I think this is the method then for flotation that is much commonly performed, okay, if I'm not mistaken. All right, and this reagent, zinc sulfate solution, it varies. The specific gravity varies, so it can be 1.18 for fresh, so we use SG 1.18 na zinc sulfate solution for fresh stool specimens, and SG 1.20 for preserved stool specimens. So how do I remember? No, kasi this is from Bailey and Scott's, which I think lumalabas din sa boards. I think this has been already asked in the board exam. Not during our time, okay? All right, how do I remember if 18, di ba? If 18, medyo fresh ka pa. All right? So if 18 years old, di ba? You're still fresh. Pero if 20 na, medyo hindi na. All right? So again, 18, 1.18 or 18 years old, you're still fresh. Okay, so 1.18 for fresh stool, 1.20 for preserved stool. Okay? All right, so 1.18. So if you're still 18 years old, fresh, like me, 18 years old, charot, joke lang. Feeler, 18 years old para mga 4 years ago. Charot, okay. Tama, 4 years ago, 18 years old. Anyway, again, mnemonic lang. 18 years old, you're still fresh. So 1.18 for fresh stool. And 1.20 for preserved stool. Okay, all right, ayan. And again, as mentioned, still it's a flotation method. So operculated eggs and heavy eggs may not be detected. So example, again, Clonorchis or unfertilized Ascaris eggs. Because again, diba, recall the appearance of your unfertilized Ascaris egg. It's elongated. It's narrow, diba? And it could be decorticated or corticated. But again, it's quite heavy. So again, it cannot float. So therefore, it cannot be recovered by a flotation method. Okay, all right. And as mentioned, to ensure detection of all possible organisms, both the surface film and the sediment should be examined, ideally. All right, so you may be thinking, sir, this is a flotation method. Why is there a sediment? Okay, or why should we examine the sediment also? Because again, there are some operculated or heavy eggs that cannot be recovered, so therefore it will sink to the sediment, okay? And this procedure, it entails the use of centrifugation. So when you centrifuge something, there will be a sediment that will form at the bottom, okay? So to ensure the recovery of the parasites that can be found or that may be found on the stool sample, then it's recommended that you examine the surface and then you examine also the sediment. But do not be confused because this is still a flotation method. It's not a sedimentation method, okay? It's still under flotation method. The general principle pa rin is that the parasites are much less dense, than the reagents. Therefore, they rise to the top. Okay, this is just a recommendation. Okay, so do not be confused. It's still a flotation method, but for the purpose of recovering all the parasites that can be found from the stool sample, then it is recommended to examine also the sediment that can be found at the bottom. Okay, but again, this is still a flotation method. All right, I hope you get that. Okay, all right. So again, here are the materials and reagents. Still the same with your brine flotation. The difference lang is, again, the reagent. 33% zinc sulfate solution. Okay. All right. And there's also a gauze, okay, that is additional, that is included. Okay. All right. Now this, from Faust in 1938, various modifications have been created with this test. So it can be used on fresh sample, yes, or formalin preserved stool samples. All right. So a lot of modifications have been done on this method. Okay. But again, it can be used on both fresh or formalin preserved stool samples. Okay. All right. Now for the procedure, the first thing that we do is we strain a well-mixed fecal suspension through one layer of gauze into a tube. So, you have a container here. Okay? Alright. So, okay, let's say this is formalin preserved stool. Okay, formalin preserved. So what you do first is, you have a receiving tube here. So you put a gauze. Sorry guys, di talaga ako marunong mag-drawing. Is this even a gauze? Para siyang hair. Alright, so you put that here. Okay. So you strain the samples, of course. Some liquid will be put here. And your stool sample. Okay. Alright, so what is the purpose of straining? To remove mga fecal debris pa rin na much larger. Okay, mga like bigger clumps, no, or bigger fecal debris. Yeah, to remove that. So we strain that using one layer of gauze. The next thing is we then centrifuge this, okay, this whole tube, and we decant the supernatant. So when you say supernatant, okay, so let's say you have already a tube that has been centrifuged. So it has now been centrifuged. So you have here the sediment, and you have also the liquid here. Okay, so this is the supernatant. Okay? Alright, diba? I know you have tried centrifuging. Supernatant is the liquid that is on top of the sediment. So we then remove this, decant. So ito ilabay. Okay? Alright? And then after removing the liquid, we then add, okay, let's say we have decanted, we then add your zinc sulfate. Okay? To 2.5 cm, so mga dito, of the rim of the tube. Okay? Until here. 2.5 cm. And then we mix thoroughly with an applicator stick. We then centrifuge again for 1.5 minutes, and then still the same, the appearance is there is a sediment at the bottom, and then you have the zinc sulfate this time, here, layer, zinc sulfate, and then you have here the surface film where the parasites will then be recovered. Okay, so we have a picture later. Okay, so again, we then transfer that to the rack, careful not to disturb again, so that the parasites will float, diba? Okay, and then let it stand for 1 minute. And then we place the cover slip pa rin here, same lang siya sa imuhang, same with the brine flotation, and then we examine the mount immediately or within 1 hour, okay, under the microscope, and report the parasite seen, the number of parasites seen. Okay, so here are your picture, ayan. So layers of the tube after centrifuging. So you have the surface, the zinc sulfate solution at the middle, and you have the sediment. Okay, and in picture, ayan. So letter A is the surface film. So you examine here, the zinc sulfate here, and lastly, the sediment. So diba, recommended that you examine both sediment and the surface film to ensure the recovery of the parasites that are found in the stool sample talaga. Alright, but again, do not be confused, this is still a flotation method. Okay, so the principle is still the same. Parasites are less dense than the fluid or the reagents used. That is why they rise to the top or they float to the top. So we recover them at the top of the preparation. Okay, so that's the zinc sulfate flotation method. Yeah, I think we will not perform this. We'll not perform this in the lab. Yeah, so at least you have a picture, okay, of the layers. All right. Okay, and the last method for flotation methods are your Sheather Sugar flotation method. Okay, now for Sheather Sugar flotation method, guys, okay, you're supposed to be, you have a manual for parasitology, diba? And supposed to be under face-to-face classes. You have the manual, and the manual created by Ma'am Bernal is really beautiful. It's really very complete and very organized. So this is not necessarily supposed to be ang ating mga ang PowerPoint, but because again of online classes, so I made a PowerPoint, and all the information, most of the information, I got it from the manual of Ma'am Bernal for parasitology lab. And for Sheather Sugar, there's no procedure, okay? So just mga information lang. All right? So again, that is why walang procedure dito. I just included additional information na lang. All right, so first is, by the name itself, Sheather Sugar. It uses sugar boiled in phenol, okay, with an SG of 1.27. So it's really high, diba? Okay. And it's considered best for your Coccidian oocysts, mainly Cryptosporidium spp. Now, if you can recall in our virtual microscope activity, diba, I've shown you one Coccidians, diba? So your Coccidians are under the sporozoa na group sa ating mga protozoa, or your apicomplexans. You'll have more of that in your lecture. So, same group sila with your Plasmodium, di ba? Species. Okay, so those are Coccidians. And usually they target the intestinal Coccidians. And aside from that, can you recall pa sa virtual microscope, we have 3 major genus and species. You have Cryptosporidium, as mentioned. Cryptosporidium. You have Isospora, or I think ang new name niya is Isospora. And lastly, you have Cyclospora. Okay, Cyclospora. So those are different, ha? 3 main genus, and I think Sarcocystis pala. Sarcocystis. Okay. These are the main genus under your Coccidians. Okay? Alright. Now, these intestinal Coccidians, by the name itself, they attack your intestines. Alright? And generally, aside from that, ano pa nga yung na-mention ko sa virtual microscope activity, they are partially acid-fast. I'm not sure if Sarcocystis is acid-fast though. Okay, so they are partially acid-fast. Okay. All right. So what does that mean? Sige, later. We'll look into that. Okay. All right. So again, since not much information about Sheather Sugar, I just included information about Cryptosporidium spp, which is mentioned here. Again, Cryptosporidium can contain 2 species. Cryptosporidium parvum, which can infect both animals and humans, and Cryptosporidium hominis, by the name itself hominis, primarily humans ang kanyang ini-infect. So what is the infective stage? Your oocysts. Okay, so that's a type of cyst, actually. That is part of the life cycle of your Coccidians, okay? Oocysts. And the mode of transmission is ingestion of contaminated food and water which contains the oocysts. Okay, so quite the same, but in fecal oral. And the disease that it causes is known as cryptosporidiosis, all right, which is common again in elderly, and as I've mentioned, in our virtual microscope activity, usually in immunocompromised patients. So, sino yan sila? Of course, you have mga AIDS patients with AIDS. You have also mga immunodeficiency syndromes. Dito mababasa. Okay. Yes, Mark. Okay. Alright. Sige, erase muna tayo. Yes. Okay. Alright. So, again, immunocompromised. Example, AIDS patients. Sino pa? You have also mga patients with immune system diseases like mga cancer of the immune system, mga immunodeficiency states. Example, immunodeficiency states. Example, like there's something wrong with its immune system, like low T cells, no? Low T cells, low B cells, okay? All right, something like that. More of that in your IS in the 3rd year. So, I don't want to elaborate further kay basin matiyam ay mo, okay? All right, or mga gikan og surgery, those after surgery, because again, their immune system is quite down, di ba, after surgery. So, they're very much prone to infections. Ayan. All right. And aside from that, they have also been implicated, as I've mentioned in your virtual microscope activity, if you can remember, they have been implicated in waterborne outbreaks of diarrheal diseases. As I mentioned, if you watch Monsters Inside Me, which is a TV show in Discovery Channel, wow, na-promote, joke lang. I don't know if napa-Japon siya. But Monsters Inside Me, they focus on parasites good. Not only parasites, but I think fungi din yata, but mostly parasites. So those that can infect humans. So Monsters Inside Me. Very good show, guys. If you really like para, like me, very good show. Real life cases talaga. Like you can, like I realized there na parang, no, it happens in real life pala, no? Like all of these that I'm studying, mga worms and protozoa, whatever, they happen in real life. Yes. So amin ako like kutob rin siya o like study. No, it happens in real life talaga. So that's a good manifestation of that. Monsters Inside Me. And there was a case of cryptosporidiosis, outbreak of cryptosporidiosis, Cryptosporidium parvum. And the first case was, the start of the episode, it focused on an AIDS patient. Okay, on an AIDS patient, so immunocompromised patient. And then, nag-start siya og kalibanga, no, diarrhea and all that. And then eventually, the patient died, unfortunately, because again, immunocompromised. So, yeah, but under normal, no, healthy individuals, cryptosporidium, usually, if you have infections with cryptosporidium, it's normal, and diarrhea, no, abdominal pain. Okay, so I think it's self-limiting. You can recover, diba? Through bed rest, fluid replacement, and all that. But in immunocompromised patients, elderly, immune system is quite low, no, quite down na. So the effects are quite severe. Okay? So yes, dami kong chika. That's for cryptosporidiosis na disease. Really good show, guys. Promise, I recommend. Okay, in your free time. If my free time pa. Charot. Okay. All right. Ayan. And again, as mentioned, it's modified acid-fast stain positive. So it appears color red or purple. Okay. Now, what do you mean by acid-fast again? Acid-fast, in the cell wall of the parasite, they have specific components that can capture, okay, or that can retain, meaning pwede nilang they can retain, no? They can hold on to the stain even after adding acid. Okay, that is why parang it's called acid-fast. And the true acid-fast, diba, again, as mentioned sa ating activity, sa ating activity on virtual microscope, the true acid-fast good is for your Mycobacterium tuberculosis detection, okay? Or this is the true acid-fast organism. Mycobacterium species, specifically Mycobacterium tuberculosis. And the acid-fast stain procedure is primarily used for the detection of MTB, which is the causative agent of your tuberculosis. So this organism talaga ang true acid-fast. Cryptosporidium, Coccidia, they are all partial lang, okay? All right, so not really true acid-fast, okay? All right, so I think we'll discuss that in the staining of fecal smears, all right? And you'll discuss this in your third year in bacteriology, okay? All right, ayan. Okay, all right, so these are your Cryptosporidium unstained. So as you can see, it's quite difficult to identify, no? They look like WBCs naman. And that's why they're very much easier to identify if you put stain on them. So this is now their appearance. Again, bright red or purple ang color nila. Because again, it's partially acid-fast. So these are the oocysts of your Cryptosporidium, di ba? Oocyst. So if mo agree ka sa imong friend, oocyst, charot. Hi, okay, Mark, wala. Ako na batin akong joke, kaya wala akong joke this sa lecture. Okay, oocysts, ayan. So a short discussion lang of the life cycle of Cryptosporidium so that you will understand. Again, how do we get Cryptosporidium? We get it through food and water, usually contaminated with the oocysts. Okay? All right. And usually, your cows, are major hosts of Cryptosporidium parvum. So example, malibang ang cow, contains oocysts of Cryptosporidium parvum. It then contaminates our water system, then it can cause infection. Okay? All right. Now, for oocyst, here, guys, no, it's quite similar with your life cycle with Plasmodium. So I will not I will not elaborate further. Okay? So we'll have a different discussion on life cycle of Plasmodium when we go to blood specimens for parasite examination. I just have to note that your oocysts, for oocysts of Cryptosporidium, guys, generally, the oocysts of Coccidia, they contain a sporocyst, and inside the sporocyst is a sporozoite. Okay? Sporozoites. Okay. Generally, for the Coccidians, so Cystoisospora, Cyclospora. But for Cryptosporidium, very characteristic niya is, for Cryptosporidium, wala na siya, okay? Its oocysts do not contain the sporocyst. So it does not have a sporocyst, okay? But it already contains directly the sporozoites, okay? All right. That's just a very characteristic of Cryptosporidium. So what is an oocyst? So example, this is the appearance of oocyst. So inside pa talaga, there's an inner shell, okay, that looks like para mga circles. So example, this is now your sporocysts. And inside, mga small dyan, those are now your sporozoites. Okay? All right. And these sporozoites are the one that matures. Okay? That is part of the life cycle. Okay? All right. So this is the general structure of the other Coccidians aside from Cryptosporidium. Because for Cryptosporidium, so this is the oocyst. All right? Ang sulod niya contains already four sporozoites. Okay? So again, it does not have any more sporocysts. Diretso ng sporozoites. Okay? Alright, I hope you got that. That's just additional. Okay? Alright, so this is the life cycle so that you can get it. Okay? So again, I'm not elaborating much. But again, from oocysts, it matures into the different merozoites, sporozoites, and then your gametes. Okay? So again, those are just, again, the life cycle of your cryptosporidium. Okay? And then you'll have that in your lecture pa rin. Okay? Alright. Okay, so we continue now. I think for the next video na ni. Yes. For the next video, we'll continue now with the last type of your concentration techniques, which are your sedimentation techniques. Okay, so I'll see you on the next video. Alright.